Good evening, everyone. And today I'll be talking about the two of the technologies which are available at Bioprocess Engineering Laboratory of CSIR-NCL. So they are like Ranibizumab and Anakinra, as well as the other technologies which are in the pipeline. I'm Dr. Rahul. I'm currently working as a senior scientist at CSIR-National Chemical Laboratory, and I lead a group on Bioprocess Engineering at CSIR-NCL. So here is the outline of the today's presentation. So I'll be first talking about the CSIR-National Chemical Laboratory, then introduction to my group, like Bioprocess Engineering Group. Then I'll be talking about the technology 1, which is a Ranibizumab. Then we'll be discussing about the updates on the technology 2, which is Anakinra. Then I will be discussing about other capabilities and offering, and finally the next step. So moving on to the first slide about CSIR-National Chemical Laboratory. So the National Chemical Laboratory is a constituent lab of CSIR, Council of Scientific and Industrial Research India, which is located in Pune, with a focus on chemical, materials, biological sciences, and engineering. NCL holds a track record of the technology transfer. More than 200 technologies are transferred till this time, and we are closely working with the industry, which includes mainly Fortune 500 as well as Sensex 50 companies. We offer the attractive models of the engagement and flexible terms for the IP for the process development as well as the technology transfer. CSIR-NCL is a publicly funded non-profit R&D lab and also has a DSI recognition called CIRO. NCL holds a long list of the meritorious partnerships with the industrial clients, which range from the short-term to the long-term engagement. With this introduction, as I told, I'm Dr. Rahul. I'm working as a senior scientist and leading a group in bioprocess engineering. So mainly we work onto the applied protein biophysics as well as the translational research in biopharmaceutical manufacturing, specifically upstream and downstream process development. So my current team strength is close to around 6, and we do have the well-equipped labs and analytical facilities, which include mainly continuous processing platform for the monoclonal antibody therapeutics, high resolution and high definition mass spectrophotometer. So what we do at Bioprocess Engineering Group. So we are an unique initiative to provide the solution for various process as well as the product related challenges in various recombinant protein manufacturing. We are mainly engaged into a joint process development project with industry to provide the de-risked intellectual property-based processes. We do the collaborative projects for the structural and functional characterization of various biosimilar recombinant proteins as well as the innovative recombinant proteins. Then doing the consultative services for the bioprocess optimization as well as the analytics. And finally, we do the applied skill development for various postgraduates and industrial resources. So here are a few of our current industrial collaborations and the sponsors of the project, where we mainly work for various types of the biosimilar recombinant proteins. So moving on to the infrastructure and then the facilities which are available at Bioprocess Engineering Laboratory at CSIR-NCL. So we are well equipped in all the instrumentation in upstream, downstream, as well as the analytical characterization for the recombinant proteins. So mainly in upstream facility, we have a high throughput microfermentation system, bioreactors of the varying capacities up to 10-liter scale, and all the types of the molecular biology equipment. In case of a downstream, we are very well equipped with the chromatography instrumentation till pilot scale, which also includes the clean column assembly system for the continuous purification of therapeutic proteins, conventional assembly systems, then CSF for the ultrafiltration, diafiltration, as well as the nanofiltration of various process flow streams. In analytical characterization, we have all the instrumentation which is required for the characterization of a primary to quaternary structures of the recombinant proteins, and then the quantification of various impurities as well as the product itself, which includes like a conventional HPLC system for all different types of the detectors like RI, PDS, and FL. On formulation side, we have a Optimax workstation along with the FDRM for the real-time characterization of the crystallization of the therapeutic proteins, a lyophilizer, and as well as a spray dryer. So a few glimpses of the selected facility: a bioreactor up to 10-liter scale, a micro fermentation and a high throughput fermentation system from bioreactors. Then this is into the downstream processing side. This is a continuous chromatography machine, so we can connect the two columns at the same time, and we will have a kind of a clean column assembly using this type of a chromatography system. ACTA purifiers, two chromatography continuous systems in order to connect entire MAP platform from capture to the polishing step, so that all the chromatography columns can be connected in the series, and we will have a kind of a high throughput downstream process development for the MAP therapeutic products. Moving on to the analytical side, as I said it, like we have the different types of the HPLC with all different types of the analytical detector facilities, and then the high resolution, high definition mass spectrometer from the water. So with this introduction, I'll be moving on to the technology 1, which is Ranibizumab. Why antibody fragments and what are the antibody fragments? So the Fab is a multi-domain protein which are generally consisting of the heavy chains and then the light chain. So light chain is again composed of the variable chain domains as well as the constant domains. Currently, there are close to around 8 Fab molecules which are approved by the US FDA, 6 of which are produced mainly into the E. coli as a host cell system. Ranibizumab is one of that particular molecule. So there are also 2 other antibody fragments which are currently produced into the mammalian host cell system. So why antibody fragments and why not monoclonal antibodies? So this antibody fragments do offer the advantages such as easy penetration inside the tissues, elimination of the immunogenicity due to lack of Fc region, and critically they can be expressed using a bacterial expression system which offers the time as well as the cost-effective high throughput manufacturing process for making these fragments as compared to the MAP production which is using the mammalian cell culture system. Along with this cost, there are the certain cons associated with the use of the antibody fragments, which mainly includes the reduced stability of the fragments compared to the full-length antibodies, short circulation half-life. That's why sometimes these are the pegylated in order to improve the circulation half-life time of this antibody fragment. And a critical bottleneck from the manufacturing side is the requirement of an efficient in vitro refolding process. Because most of the time, although we express it cytoplasmically or the periplasmically, they lead to the formation of the infusion bodies. So the molecule what we have selected for this study or what we are discussing is the Ranibizumab. This is a recombinant humanized IgG1 monoclonal antibody fragment which acts against the VEGF-A specifically. And the innovator molecule is Lucentis, which is marketed by Genentech for the Novartis, which is approved by US FDA in June 2006 and by EMA in January 2007. So the patents are near expiry in Europe, whereas they are already expired in US in June 2020. So as I said it earlier, this is a Fab molecule which is having a molecular mass of 48.380 kilodalton. There is a heavy chain and then there is a light chain. The complexity is about the total 5-disulfide bond formation which is required for making this molecule. The 2 are the intra chain in light chain, 2 are the intra chain in the heavy chain, and then 1 interchain between the light chain and the heavy chain. The indications which is Ranibizumab is given is majorly age-related macular degeneration and diabetic retinopathy. What is the market and industry overview for this particular molecule? As I said it earlier, the global age-related macular degeneration market is really growing fast, and it is up to valuation close to around $1.58 billion in 2020, whereas it is expected to reach close to around $2.64 billion by the end of 2026, which is growing at the rate of the CAGR of 8.93%. So there are 2 global players, mainly Genentech and the Novartis, whereas in India there is only 1 player, which is Inta, which is making a biosimilar version of the Ranibizumab. So where is the opportunity? So AMD generally is affecting close to around 7 to 8% of the worldwide population, and number is projected to increase around 196 million in 2020. So projected number of the people with this is around 196 million in 2020, which might increase to close to around 300 million in 2040. Cost is still high for this particular product. So 50% of the patient onto the VEG therapy drop out of the therapy. So they generally are the dropout after the initial injections. So because of the non-affordability of this particular injection, and still there is a chance like we can really make this molecule economically affordable to the population specifically for the Indian population. So industry is not yet crowded. There is only first ever biosimilar which is available from Intas with the title Razumab, which was launched by Intas Pharma 2015. And there are also the new indications. Now currently it is also recommended for the use in a diabetic retinopathy. So this can really help us in decreasing the cost of this particular product. So what we are going to offer, what is our technology offering is? So we are going to offer a clone, upstream, and then the downstream process for making this recombinant human Ranibizumab, which is a biosimilar version of a Lucentis. What are the advantages we are offering over the existing processes which are also making the Ranibizumab? So in case of the upstream, we have the single fermentation batch. So we have a duet expression system which expresses both light chain and then the heavy chain for the making this Ranibizumab. The original biosimilar which was launched by Intas has the 2 different fermentation strategies where they make the light chain in one host in one fermentation batch, whereas the heavy chain in another fermentation batch. So with the use of the duet expression system, this particular disadvantage can be removed. Then we'll be offering a high throughput refolding process. For this particular complex molecule, the currently in the reported industrial yields of this refolding process, they are very low, less than close to around 15%. So our current refolding strategy gives close to around 40 to 45% of the refolding yields. So we have designed a purification process which involves the combination of the two multimodal chromatography steps. So these are the salt tolerant chromatography in an anion exchanger as well as in the cation exchanger mode, which offers close to around 2x improvement into the overall process productivity. The purification platform which we are applying, so that is also applicable for the solubly expressed antibody fragment. And as I said, we are using a multimodal chromatography which overcomes the advantage of existing affinity mode separation for this antibody fragment, which is the cost center in the downstream processing of antibody fragment. So this reduces the further cost to 1/3 by replacement of this affinity chromatography with use of a multimodal chromatography. So we have two different patent filings for making refolded Ranibizumab and then the purification of the Ranibizumab. Currently, they are in application stage in various countries. So so this was all about the details of the technology transfer, which we will be interested in transferring. So let's see how does the data look like. So first is the upstream data. So as we know the amino acid sequence, we started with the codon optimization. So as I said we have a duet vector which is expressing the light chain and the heavy chain of the Ranibizumab. So then this vector was transformed into the host cell E. coli BL21D3, which is already a off-patent vector host cell system, then into the high cell density fermentation leading to formation of infusion bodies, and finally solubilization and in vitro refolding of the Ranibizumab. So how does this look like at the expression level? So the LEN1 is the expressed light chain and the heavy chain for the NCL clone, whereas the LEN3 of this SDS page is indicating the lucentis under release and alkylated conditions. Okay, which is separating the heavy chain and the light chain of the Ranibizumab. So after in vitro dilution based refolding, how does the molecule looks like? So again, onto this SDS page, the second lane is the originator innovator molecule lucentis, whereas the third line is indicating a refolded Ranibizumab into the NCL process, which is indicating a 48 kilodalton molecule is formed after the in vitro dilution. So as I said it earlier, the in vitro refolding is the critical bottleneck. So where are we with respect to the existing industrial standards and then the existing patents? So here I have listed the existing reported yields of this refolding for various patents from Intact, which is claiming close to around 9% of the refolding yield in 120 hours, whereas the Lupin is claiming close to around 15% of the refolding yield in 72 hours. And then the Rudolph, the original investigation which was published in Nature, claiming close to around 40% of the refolding in 120 hours. So this field boxes are indicating the refolding yield in a current investigation, which is close to around 50% in 120 hours. But we are stopping close to around 72 hours, which is giving us the refolding yield of between 30 to 40%. So this is the typical advantage which is offered over the conventional existing refolding process. So another data on the batch consistency. So here is the batch consistency data for 3 cyclical batches, which is indicating a total protein yield, including light chain and heavy chain, close to around 1.5 grams in a batch fermentation, whereas the build yield will improve to close to around 3 grams per liter in case of fed batch fermentation. And here is the SDS spread, again indicating the consistency into the profile and comparison to that of the lucentis. Moving on to the downstream processing platform, as I said it, like we have used the two-stage multimodal chromatography platform followed by the UF and DF of a refold, which helps us in capture as well as the polishing of this particular antibody fragment. So here are the chromatograms for this multimodal chromatography, and then the SDS page indicating a comparative analysis with the Novartis lucentis versus the NCL lucentis. So they exactly match with their profile. So now let's come to the analytical data. How does the intact mass analysis look like for this intact molecule and then at the subunit level? So the innovator molecule showing an intact mass lucentis of a 48379.713, with a biosimilar version of that is showing the exact matching molecular masses of 48379.719 daltons. So let's see what happens at the subunit level. So when under lucentis under reduced and alkylated condition, the light chain gives a molecular mass of 23428.596. Whereas in case of a biosimilar, this is close to around 23428.773. And then in same case, in the case of the heavy chain, so again both are indicating an exact match with the lucentis. So further to this, we have also confirmed the purity based on to the RPHPLC and the SEC analysis, indicating the absence of the aggregate, as well as the RPHPLC method was used for the quantification. The interesting thing was checking the disulfide whether they are at the appropriate position. So in order to do that, we have done the two-phase analysis under reduction condition as well as non-reducing condition, which are orthogonal enzymatic digest like trypsin and gluci plus chymotrypsin. And we have used two different types of the mass spectroscopy platforms like a TOF and then the trap-based system. So in case of a TOF, we have used the CID plus ETD-based orthogonal fragmentation, whereas in case of the orbit clip, we have used the STD-based fragmentation. So how does the result look like? So specifically, I am just showing the interchain disulfide bond formation, specifically like how it looks like. So here is the light chain as well as the heavy chain, and this is the interchain disulfide bond. So as I said it, like we have confirmed both orthogonally D ions, Y ions, as well as the C ions and the Z ions. Okay, so all the fragments were observed and confirming to the monoisotopic mass of the 77.5.3886. So that confirmed the presence of the interchain disulfide bond. So the type of the test we have conducted at NCL to confirm the biosimilarity is mainly molecular mass, primary structure, secondary structure, aggregate quantification, STPs, residual DNA, amino acid sequence, disulfide bond mapping, and we are not completed with the pyrogenic testing. And we have observed that all the analysis is matching with the in-vitro molecule. So what is the current stage of the development? We have done the scale up to 10-liter reactor with the 3 consistency batches for it. 5 consistency batches at the 10-liter scale, and current yield is close to around 2.81 grams plus minus 0.1 gram per liter. So we have scaled up this process, and now we are ready to generate the material for the preclinical studies. So that is the current status of this particular technology. So moving on to the technology 2, which is the anakinra. What is anakinra? So anakinra is a recombinant non-glycosylated form of the human interleukin-1 receptor antagonist that reduces the activity of interleukin-1, which is the key driver for the inflammation in autoimmune and autoinflammatory diseases. So this is particularly given in the treatment of the rheumatoid arthritis as well as the Still's disease, neonatal onset of a multisystem inflammatory disease, then cryopyrin-associated periodic syndrome, and familial Mediterranean fever. So these are the indications currently approved by US FDA. So originally this drug was approved in 2001 and by EMA in 2002, and then the original patent was expired in 2008. So now this is off patent. So why you should be interested in this particular molecule? So nearly 4% of the world population is affected by one of more than 80 different autoimmune diseases like rheumatoid arthritis, and then the most common disease is the rheumatoid arthritis. There are a lot of the treatments available for the rheumatoid arthritis, but the cheapest version is available with the anakinra. So there is a global prevalence of rheumatoid arthritis is between 0.24 to 1%, and then there is a new indication for this. Okay, so if we look at the cost for the anakinra, as I said, this is the cheapest treatment which is available for the rheumatoid arthritis, which is offering the treatment at the 1/10 of that of the rituximab, which is a mAb which is produced using the mammalian cell culture system. So currently this is also having a novel application in COVID-19 induced SARS. So the clinical trials are currently ongoing, and then the results will be declared close to around October 2021 for the use of this anakinra in COVID-19 induced SARS. So industry is not at all crowded. There is only one company which is making this particular molecule, and cost is still high, close to around $1,000 for 4.69 ml of a single dose. So opportunities for the process innovation and the cost reduction is possible for this particular molecule. So moving on to a technology offering, what we are offering for this, again a clone, upstream, and then a downstream process for making anakinra. So here we are not offering a kind of a refolding based process, but we are offering an advantageous like soluble expression for the anakinra, which eliminates a completely an in-vitro refolding step. So there is a purification process which involves novel multimodal chromatographic purification, again improving the product overall process productivity by 2x as compared to the conventional cation exchangers and anion exchangers. So time effective and cost effective process, since we eliminate completely the in vitro refolding of this particular process. And currently the yield is close to around 1 gram per liter of the fermentation broth. So again, similar to that of the first molecule, Ranibizumab, we know the amino acid sequence for this particular anakinra. So we started with the codon optimization, then went for the vector construction transformation into the host cell E. coli 2023, high cell density fermentation, soluble expression, cell lysis, and finally the separation of the cell mass and the solubilized anakinra. So this is the typically the express molecule, and then the clone details how it looks like. Again, similar to the Ranibizumab process, we have the two-step multimodal chromatographic purification for the complete purification of the anakinra. So how does the biosimilarity look like for this molecule? So again, we have done the ESI-MS analysis for the innovator, which is the kinerict, and then the biosimilar version of the NCL molecule. So the observed mass is 17254.67 in case of our molecule, whereas 17256.97 in case of an actual kinerict. The mass error is already indicated over here, which is within limit of proving the biosimilarity for the molecules. Then after confirming the molecular mass at the intact mass level, we went for the peptide mapping, where we have done the orthogonal enzymatic digest using different types of enzymes like a trypsin plus gluci and then the chymotrypsin. And then we have checked for the peptide fingerprinting of the innovator versus the biosimilar version. So now what we have seen is close to around 100% of the sequence coverage was observed for this particular molecule as well. So this was again confirmed at the MS2 level. So the first one was at the MS level, and then we have further fragmented both innovators as well as the biosimilar versions and cross-checked the further fragmentation at the MS2 level and confirmed that all the fragments which were observed at the MS were further fragmented into MS level, confirming the biosimilarities with that of the kinerict. So what are the tests we have currently performed and what we are going to do in the future? So mainly we have performed the molecular weight confirmation for this particular molecule using SDS-8, Margitox, SEX, ESI-MS. Secondary structure, carbohydrate content is not applicable for this molecule. Then aggregate quantification, SCP, residual DNA, and disulfide bond mapping using LC-MS and amino acid sequence. So completed all the biosimilarity analysis which is required for at least for the RCGM submissions, and we have observed the good agreement between the innovator and the developed biosimilar protein. So similar to the first molecule, we have done the scale up for this particular molecule at the 10-liter scale, and currently it is close to around 1.1 grams per liter of this molecule. And we are currently in the process of doing the consistency batches and then further path will be to generate the material which is required for the preclinical and then toxicity study. So with this, I complete the presentation onto the technology showcase. I'll just show the other molecules which are there in the pipeline. So we are working with the romiplastin, for which is the molecule given in the treatment of ITP. We have completed the clone and the upstream process development, and analytical characterization and downstream process development is under progress. The other molecule is the teriparatide, which is given in osteoporosis. Then we are working with the GCSF, which is given in neutropenia. Insulin Vispro and L-asparginase, we are working in collaboration with IIT Delhi with Professor Anurag Rathore. And for that, we have completed the clone and the upstream process development, and currently we are developing the downstream process as well as the analytical characterization of this molecule. What will be the next steps and what we are seeking from the industrial partners? So we are critically keen to forge the industrial partnership for specifically advancing the biosimilar technologies which are presented today through in vivo or the clinical studies, and we look forward for licensing of this technology know-how with patents, joint development and technology advancement and scale up of this particular project. Sponsor project for the process development for other biopharmaceutical industrial projects utilizing our expertise, capabilities and facilities with the group. And then finally the consulting project.